Normally we act this out together in class. Everybody gets involved and it's a whole lot of fun. I hate that you're missing it, and so I'm going to try to do all the parts myself. It may get a little ridiculous, but we're going to give it a try. So what we're looking at here is a representation of a cell. In green, this strap here represents the plasma membrane of the cell. Here, there is a white wire that's coiled to represent the 7 alpha helices that pass through the membrane. It's a G protein coupled receptor, and it has these 7 transmembrane alpha helices that pass through and form the structure. The GPCR is represented here. Here we have our G protein. This is a trimeric G protein. It's not exactly like the RAN and the RAS and the others that we've talked about up to this point. This one has 3 components: an alpha component where the GTP or GDP combine, a beta subunit here in the middle, and a gamma subunit at the other side. Both the alpha and the gamma subunits are lipid linked into the membrane, which is represented by these green pipe cleaners. The beta subunit is affiliated with the gamma subunit here. In its inactive state, it's bound to GDP, and it sits quietly in the membrane. The G protein coupled receptor does not have a signaling molecule to it, so it's just sitting quietly in the membrane as well. There are other enzymes present in the membrane along with these components. Adenylyl cyclase is one that's going to be important in our story, so I put it in here. We could also have a potassium channel or something like that. So inside the cell, of course, there are a lot of things present in the cell, but the things of interest to us inside the cell are ATP molecules and another enzyme that's called phosphodiesterase. Phosphodiesterase is a water-soluble cytosolic protein. So we have a clip right here. This clip is going to represent our signaling molecule. We'll use adrenaline as our signaling molecule. Adrenaline does activate G proteins very effectively, and so our adrenaline is secreted from the adrenal gland. It circulates in the bloodstream, it circulates through the extracellular space, and as it encounters a cell with a G protein coupled receptor that is complementary to it, it will bind in its binding site onto that receptor. When it binds to the receptor, it activates the receptor. The receptor in this bound state is now capable of calling the G protein to itself. An activated GPCR will draw the G protein to itself, and it will instigate activation of the G protein. So what activates a G protein? Binding to GDP. So the GDP is released, a GTP is bound, and now we have an active trimeric G protein. The trimeric G protein tends to, upon being activated, separate so that the beta gamma subunit will go do one thing and the alpha subunit will go do something else. I couldn't make this three-dimensional, so we're going to pretend that it is, but we want this alpha subunit to interact with an enzyme such as our adenylyl cyclase here. When it interacts with the adenylyl cyclase, as you can see here, GTP, the adenylyl cyclase, it'll activate the adenylyl cyclase. When it activates the adenylyl cyclase, the enzyme will take ATP molecules that are present in the cell, and it will cause them to break so that now it's an adenosine monophosphate. Not just an adenosine monophosphate, but it is a cyclic adenosine monophosphate. In its cyclic form, it can go perform various functions in the cell. So we'll take this one and let it go do some things in the cell. We'll take another one here. It will come to the adenylyl cyclase. lose 2 of its 3 phosphates. It'll form a cyclic AMP. In the form of cyclic AMP, it will go do other jobs in the cell. Cyclic AMP. And it will continue. We have other ones here as well. And so our cyclic AMP, written lowercase c, capital AMP, is one of the most important second messengers that cells use inside. So the signaling molecule, remember, the adrenaline can't actually make its way into the cytosol. So instead, it transmits its signal and it's transduced into a different kind of a signal. By activating our G protein, our trimeric G protein, then the activated G protein can cause the production of the second messenger, these cyclic AMP molecules. As they go and activate various responses within the cell, more and more will continue to be produced as long as GTP remains bound to the alpha subunit. Over here, our beta gamma subunit will most likely interact with some other membrane-bound enzyme, like, for example, an ion channel that's responsive to beta gamma binding to it. It could open the ion channel and allow potassium, for example, to rush out of the cell. So eventually, the alpha subunit will hydrolyze its GTP. By hydrolyzing it, that means it's going to gradually remove its 3rd phosphate group, and so the GTP will convert to GDP. When it converts to GDP, it's no longer active. It can't interact with the adenylyl cyclase anymore. It can't cause any more production of cyclic AMP molecules, and instead, it will just return to its original position where it will interact with the beta gamma subunit, and as long as adrenaline is bound to the G protein coupled receptor, although it could activate other copies of the trimeric G protein, at this point we'll say that our adrenaline is released. It doesn't stay bound long, but it'll bind and release. We'll say our adrenaline is released, and so the signal is stopped as far as the membrane events are concerned. However, as long as we have cyclic AMP inside the cell, it's going to keep on causing the response to the adrenaline. Therefore, we need to stop this cyclic AMP response. That's where phosphodiesterase comes into the story. Phosphodiesterase cuts the bond that causes the cyclic part of cyclic AMP. It basically releases one part of the phosphate group from the stem of the AMP. So it's just simple AMP rather than cyclic AMP. It'll do the same thing here. And in this form, they're no longer active. And so we've inactivated our receptor by the loss of adrenaline. We've inactivated our trimeric G protein by the conversion of GTP to GDP. We've inactivated our adenylyl cyclase because it's no longer being stimulated by the alpha subunit. And we've inactivated our second messengers, the cyclic AMP, by the activity of the enzyme phosphodiesterase. That's the story.